So this lesson is focused on enzymes. Enzymes are proteins that drive change. And let's look at this image to explain what is the role of an enzyme. So over here we have a sucrose, a disaccharide, and in presence of the enzyme sucrase, we are able to break down. So the hydrolysis of the sucrose happens in presence of sucrase, the enzyme, and it breaks into glucose and fructose. So that's a classic reaction that could be happening in your body right after lunch or when you had candy or chocolate. So a catalyst is a chemical agent that speeds up a reaction without being consumed by the reaction, and enzyme is a catalytic protein. Hydrolysis of sucrose by enzyme sucrase is an example of enzyme catalyzed reaction, as we just looked at this image. Next image. So for free energy and a little bit more about that, watch my free energy video. So essentially, if you have a diagram where you have a graph of progress of a reaction and free energy, you will see that the reaction proceeds towards decreasing the free energy, right? In this case, this reaction is proceeding towards decreasing the free energy. And A, B, C, D, those represent unique atoms that are bound to each other with a certain bond energy. So these reactants at elevated energy level and with the threshold energy being applied, they go into a transition state where these bonds are no longer stable. And at that time, the evaluation of the bond energy happens, which in this case favors lower free energy. So then you get A and C combined, B and D combined. That's a typical reaction. Every chemical reaction between molecules involves bond breaking, bond forming. Initial energy needed to start a chemical reaction is called the free energy of activation or activation energy. Activation energy is always supplied in the form of heat. So it could be heat or energy that would be supplying that free energy. Enzyme catalyzes reactions by lowering the energy barrier. Enzymes do not change the free energy, delta G. Instead, they hasten the reaction or make it faster that they can occur. And in the video, we talked about the example of what is a free energy. It really represents stability. So it's saying the trolley on the top of the hill is less stable than trolley at the bottom of the hill. And by nature, the trolley at the top of the hill is going to go down. So watch that video. And there's a similar one for bond energy. So essentially what we're saying is the reactant and the product, we're not changing anything there. The beginning and the end is exactly the same. The only effect of enzymes is it is decreasing this threshold. So it is making it easier for the reactants to come together. Okay? That is the biggest role of the enzyme catalysts. So the reactant that an enzyme acts on is called the substrate. And the enzyme binds to the substrate to form the enzyme substrate complex. How imaginative. Now, where do they bind? They bind at the active site. Active site is the region of the enzyme where the substrate binds. And because of the binding, it enhances, in the three-dimensional state, it enhances the interaction between the substrates. For example, this is the enzyme. The active site is the physical location where the substrate is uniquely positioned, is uniquely matched to work with. So as soon as the enzyme and substrate get into a complex, you will see there is a change in the three-dimensional, tertiary, and the quaternary structure of the enzyme. In this case, we're talking about the protein. It changes, and the physical change actually helps bring down the activation energy. Here is a beautiful example. So imagine this is an enzyme. It has its active site very uniquely defined to attract two substrates. So when the substrates bind and they form the enzyme substrate complex, this is a completely different structure than what you had the plain enzyme. This conformation and the binding then helps create the chemical bonds. Okay, so hydrogen bonds, ionic bonds are formed or changed. So active sites can lower the activation energy. They speed up the reaction. They provide favorable environment. They maintain the pH. They maintain the temperature, etc., so that the reaction can go forward in a systematic way. At the end of the reaction, the products are released and the enzyme goes back to its virgin shape, its original shape.